had a very nice introduction, and we saw how the brain works with fMRI and EEG. And I'm very happy to invite Dr. Tzipi Horowitz, who did a doctorate here in Haifa and has moved to Cincinnati, but we hope she'll be back soon. And she'll be able to show us with fMRI and DRP if it's possible to train the dyslexic brain to read better.
Thank you very much. Thank you, Shelley. One second. Okay, I will first like to thank the organizing committee for inviting me to give this talk. As Shelley said, as Dr. Shaul said, I did my PhD here in the Edmund J. Safra Center, so I grew with the center, and in this opportunity I would like to thank the foundation for that. Today in this talk, due to time constraints, I'm going to focus specifically on dyslexic readers and the effect of the Reading Acceleration Program on executive functions and reading neural circuits in children with dyslexia. I will give a short introduction on the relations between reading and executive functions, as well as show some of our old data using EEG and the Reading Acceleration Program. I apologize if I'm going to call it RAP, because that's how we call it in Cincinnati, so it's really implemented. But I will try to call it the Reading Acceleration Program. I will then move on. I can call it RAP.
Now have RAPI.
Yeah, RAPI. Oh, right. Okay, so we will then move on to our functional MRI data that we recruited in Cincinnati. So there are several causes and theories that were raised in order to explain dyslexia, and thanks to Danny and Ken yesterday, I wouldn't elaborate on the different causes. But I would like to draw attention to the large body of evidence in the past few years that was published on the executive deficit in children and adults with dyslexia. So the relations between reading and executive functions can be demonstrated in this pyramid here. Oh, the contrast is not so good. Do you see it okay? Whereas reading comprehension is basically at the top of the pyramid. It relies on fluent reading, on more basic language component, and at the basis of the pyramid, we have the executive functions. And this is a figure of our one of our latest analysis, showing that in typical healthy children aged 11, there is a significant correlation between greater reading scores and greater frontal activation. And frontal activation is related to executive functions. So in our clinical observations, we noticed that readers with dyslexia do not have a constant pattern for their reading errors. Namely, they can read the same word differently each time they encounter it. So we raised the question regarding their error monitoring system. And what we found is that when a dyslexic reader makes an error, you can, I will provide a short overview about that. The error detection mechanism can be detected by EEG and ERP methodology, and we can look at a specific component that is called the error-related negativity, or the ERN, or the OOPS potential, like Tzvi likes to call it. And what we found is when dyslexic readers make error, their error-related negativity potential is significantly smaller in amplitude than the one in typical readers, and it's also correlated with their reading scores. And since we are great believers in intervention, we wanted to see whether training with RAP can improve executive functions. And recent evidence from a paper that is in press showed that training with the reading acceleration program also improved attention and working memory. So we wanted to look specifically at the error detection mechanism and to see how training with RAP will affect that. And we found that yes, training with the reading acceleration program increased the amplitude of the error-related negativity in children with dyslexia, and this improvement was also correlated with an improvement in reading performance. So we suggested that training with the reading acceleration program forces the reader to process more units, more letters in a given time, and forces somehow an orthographic processing, or improves the orthographic processing, which in turn causes an increase in the mental lexicon of these readers and improves their monitoring during reading, because now they have a representation, the right representation to compare their actual response, their actual reading response to. And in the case of a mismatch, an error potential is evoked. But what are the neural circuits underlying this mechanism? And we didn't know that back then in 2011, so I had to go to Cincinnati in order to answer this question. And this is the objective of the current talk, to examine the effect of RAP on reading and executive neural circuits in children with dyslexia using neuroimaging methods. We recruited 8 to 12 years old children, all right-handed with an average IQ, and the inclusion criteria for children with dyslexia was -1.5 standard score in standardized reading tasks such as the tower, the gourd, and the C-top. We ruled out we excluded children who had attention problems, and we also measured that during the Connors task. In this point, I think it's important to give some background, because when I came to Cincinnati in 2011, I've been told that there's no dyslexics in Ohio. I was quite surprised, because I know it's a genetic problem. It probably didn't pass this region, but apparently the dyslexia legislation, the acknowledgement for dyslexia, was accepted in Ohio at the end of 2011. So by the time I started the study, suddenly I had lots of dyslexics, lots of candidates. The tool we were using was functional MRI, and we acquired 18 children with dyslexia and 18 typical readers who went through a battery of reading and executive measures, performed a functional MRI paradigm, the lexical decision task, and went through a training of 4 weeks, five times per week, for 20 to 30 minutes per day, per session, and performed the same battery and the same lexical decision task with different stimuli. A little bit about the task. We used 30 words and 30 pseudo words in a block design task. And the instruction was to push the thumb up button whenever they see a word, and the thumb down button whenever they see a pseudo word. So let's move on to the results, to the behavioral results first. What we see here, this is the reading results. The dark gray represent the data before the training, and the light gray the data after training in dyslexics and typical readers throughout the presentation. And this is the data for the speed of reading. And this is the time that took the reader to read a silently given sentence. But we found that the dyslexic reader read significantly faster after intervention, as well as with typical readers. They read more words per 45 seconds and they improved their comprehension. We didn't find an improvement in comprehension in typical readers, probably because they started with the ceiling already. And some of the executive functions results, again, dark gray for before training and light gray after training. And we found that visual attention improved in dyslexic readers after intervention, as well as the ability to divide the attention between an auditory and a visual giving stimuli. And we also found an improvement in speed of processing measure using the naming task, as well as in working memory in dyslexic readers. So let's move on to the functional MRI data. And what you see here is words contrasted with pseudo words before and down below after intervention in the left hemisphere and in the right hemisphere in dyslexic reader. And what we found is that before intervention, there was greater activation in regions in the left hemisphere, in regions related to working memory, to phonology and silent articulation, and in the cuneus, in the visual area, and also in the middle temporal gyrus and auditory-related regions. And we kind of found a mirror image of this activation in the right hemisphere. Following intervention, we found more focal activation in the cuneus, in the visual area, as well as in the premotor, in the precentral gyrus and phonology and articulation. And there is still a signature here in the right hemisphere as well. So we wanted to look at the gain in dyslexic readers following this intervention. So we performed a pair t-test before and after intervention and correlated it with the gain in words reading. And we found the activation of the anterior cingulate cortex here, which I remind you relates to the error detection mechanism. And in order to find which area differentiates the gain in dyslexic readers from the typical readers, we performed repeated measures, ANOVA, and looked specifically at the interaction of group and test. And we found an activation of the visual word film area, the fusiform gyrus, to be more active in dyslexic readers after intervention. So, we chose this region as a seat of interest, and we were looking for other regions that are function together with this region in an analysis that's called functional connectivity. And what we found that before intervention, there was greater functional connectivity between the fusiform gyrus and the dorsolateral, the left dorsolateral prefrontal cortex, which is related to working memory and semantic retrieval, whereas after intervention, the region that was highly correlated with the fusiform gyrus was the right anterior cingulate cortex. And Danny, I think it aligns with your couple of your last slide, talking about the right frontal activation as a predictor for improvement, and I would love to discuss that afterwards. I remind you that this region was related to the improvement using our EEG data after training with the rep. So, we can train dyslexics to read better. We suggest that the lower starting point is the larger is the gain, both by executive functions and by the reading measures. Training seems to shift the activation to the left hemisphere, and increase the activation and synchronization between brain regions related to mental lexicon and error monitoring. And our results trigger us a little bit to think about laterality, since we found the right, the connectivity between the fusiform gyrus and the right anterior cingulate cortex. So we suggest two possible pathways that can explain the mechanism of the reading acceleration program. The first is through a direct effect on neural circuits relating to reading, with more focal and left lateralized activation of reading circuits that may stimulate the fusiform gyrus to process words in an orthographic manner. And this may create more stable lexicon, as we hypothesized before, and improve the ability of the dyslexic reader to detect its reading errors. Or alternatively, there might be a direct effect on the executive system, and it might train the error detection mechanism as part of the learning circuits to work more efficiently, and maybe that's why they recognize their error more efficiently, and that helps them to create a better mental lexicon. And in line with this, we might hypothesize that maybe reading process is like an orchestra, and executive functions can be either the conductor or one of the players in the orchestra. In order to find the causality, or in other words, whether executive functions or reading start this sequence, we'll have to perform, and we plan to perform, an effective connectivity analysis. We would like to see whether the effect of RAP is domain-specific or general, and we already acquired the resting state data, and we want to see how it affects the general activation without any task dependent. We want to see how an additional executive functions training, like the warm-up, affect the system and how it stimulates the executive system. And of course, we would like to examine the normalization theory and to see how different the gain from training is in dyslexic readers as compared to the gain in typical readers. I would like to thank my mentors, Professor Scott Holland, and my colleagues from Cincinnati, my coordinator, Nicole Kicino. She's not here, but she's one of the reasons that this program went so fast. And of course, Professor Bresnitz, for allowing us to use this program and to develop it even further. Thank you so much for listening.
Any questions? Yeah.
If I'm the only one, otherwise I will give my turn to somebody else.
It's your turn.
I would argue that we have a conductor, not a whole orchestra, and I'll tell you why. I think that one of the things that we found out that maybe the conductor, executive function, might synchronize between the various parts of the brain. The asynchrony theory that actually suggested that there is a delay between the graphing and the phoneme. And somehow, when we are training the RAPI, we somehow push and we eliminate the delay between the various systems that are activating reading, whether it's phonology, orthography, graphing, phonic correspondence, or the visual and the language areas later on up. And I think that it is a reciprocity somehow effect. And I think that one of what you really nicely, very nicely showed us, that the frontal lobe, executive function, either it's error detection or not, are actually getting the symbols from bottom up and top down, which I'm not sure that people will like what I'm saying, but it is an interactive process. And somebody has to amalgamate the whole thing, and somebody has to be a conductor. And I think that the frontal lobe or its executive function might be a good candidate for it.
I totally agree, and I think that also with the data that you showed yesterday, that a warm-up with a cognitive training of a couple of minutes maybe help this conductor to go up on stage, lift his arms up, and then rapping pushes him to do the right thing and to make beautiful music. So I agree.
It's a methodological question. How do you design the content on which you measure the error detection? I mean, what kind of things do you need? What is the potential for errors there? How do you design?
So in a lexical decision task, it's words and pseudo words. And readers with dyslexia, when they see words for a certain time, sometimes they decide that it's a non-word just because it's not in their mental lexicon. It might take them a while. If it was in a context, it might be easier for them to decide whether it's a word or not by means of context. But if it's an isolated word and they just have to recognize it, they have to retrieve it from somewhere, and if it's not there, they make an error. So in this design,
it's not an ecological, I mean,
in this specific, in this specific study that was
so
done.
you might want to talk with you about studying international here.
The pseudo-homophones
monitoring an actual more ecological.
in an ecological reading in sentences, in context,
Homograph.
Mm-hmm.
Sure. The next BSF, no?
Next week's
Sentence.
Yeah, I just want to comment on the connection between your ERP finding and your imaging findings. In the lab where you now work, we do fMRI, but not the ERP. And the fact that you were able to identify the physical location showing enhanced activity with fMRI that corresponds directly to the region where this ERP or the event-related negativity is occurring, I think is really a good discovery. It's showing that not only is the bold effect that reflects the brain activity indirectly is enhanced, but you also show that this corresponds to a direct measure of enhanced electrophysiologic activity. It's really convincing. It's a very nice story.
Thank you. I think it triggers us to do functional MRI EEG study, right?
Yeah, that would be even better.
Thank you.
Thank you very much. We're going to have a coffee break. And